Hello everyone. In today's video, we will learn about the famous experiment of Oswald Avery, Colin MacLeod and Maclyn McCarty, commonly known as the Avery MacLeod McCarty Experiment. This experiment showed that it is actually the DNA that is the genetic material inside the cells. Today, this information seems very obvious. However, back in 1944, it was challenging to provide experimental evidence for the same. The story begins with Frederick Griffith's famous experiment in 1928, in which he discovered the phenomenon of bacterial transformation using mice and Streptococcus pneumoniae. While searching for a vaccine to cure pneumonia, Griffith injected mice with two different strains of bacteria to test whether they develop the disease or not. The S strain is pathogenic, that is, disease causing, and has a capsule outside its cell wall. The capsule allows the cell to escape the immune responses of the host mouse. The R strain is non-pathogenic, meaning that it does not cause the disease. Surprisingly, a mixture of heat-killed S bacteria and the live R bacteria caused the mice to develop pneumonia and eventually die. Griffith concluded that the R strain bacteria must have taken up what he called a transforming principle from the heat-killed S bacteria, which allowed them to transform into smooth-coated bacteria and become virulent. What Griffith, however, could not answer was what exactly was this transforming principle? Was it a protein, was it a nucleic acid, or polysaccharide? Taking lead from Griffith's experiment, Avery and colleagues set out to discover the biochemical nature of this transforming principle that Griffith had reported. The scientific community at that time believed proteins to be the genetic material in the cell. DNA containing only 4 nucleotide bases was still considered to be too simple a molecule to carry biological information. Proteins, with their 20 different amino acids, offered far more complexity and were regarded as more likely candidates. To find out the biochemical nature of Griffith's transforming principle, Avery and colleagues set up a series of experiments. They did exactly what Griffith had done in his experiments, but with the following changes. Firstly, after heat killing the smooth strain of bacteria, the mixture was treated using detergents to break open the cells. Organic solvents were used to separate lipids and carbohydrates via centrifugation. The solution now contained the major biomolecules speculated to be the genetic material, that is, the DNA, the RNA, and the proteins. Next, the mixture was divided in 3 tubes and enzymes that specifically degraded each component were added. Thus, proteases were added in one tube to degrade proteins, RNAs to degrade RNA, and DNAs to degrade DNA. This mixture was now used separately to transform the R strains. The theory behind this experiment was that if the transforming agent was, for example, protein, the transforming agent would be destroyed in the test tube containing protease but not in the others. Thus, whatever the transforming agent was, the liquid in one of the tubes would no longer be able to transform the R bacteria into the virulent S type. Apart from these tubes, a control tube was kept in which no enzyme was added. As expected, the culture in the first tube, that is, the control tube, could transform the R bacteria into the pathogenic S type, and hence smooth colonies were seen on the culture dish. The R strain in the second tube that was incubated with cell extracts treated with proteases was also found to be transformed into the pathogenic S strain. This proved that the transforming principle is definitely not protein. Similar results were obtained in the case of cell extracts treated with RNAs. The R bacteria was still able to transform into the S type. However, when cell extracts were treated with DNAs and later incubated with the R bacteria, transformation of the bacteria could not occur. The R strain could not transform and instead appeared as rough colonies on the culture dish. This proved that it is the DNA that is responsible for transfer of genetic information. These experiments clearly showed that the transforming principle that Griffith had mentioned in his experiments was nothing but the DNA. The results of Avery and colleagues were still met with a bit of skepticism from the scientific community at that time, and many still believed protein as the carrier of the genetic information. It took another group of scientists, namely Hershey and Chase, to finally end the DNA versus protein debate through their experiments.